Hi everyone, my name is Whitney Morgan. I have a Master of Science in Bioinformatics. I've been in emergency medical services for 12 years, paramedic for a decade, and also the current interim educational support coordinator for the Emergency Medical Services Program here at Northwest College in Powell, Wyoming. However, our physical offices are in Cody, Wyoming. So we're going to do a little bit of a series on the COVID disease that is plaguing the world right now. This is intended for anyone who is curious to learn more, but especially for our students whom we don't have contact with right now, just because they're on an extended spring break and also practicing social distancing. So as with any of my classes, we will start with a little music to get our brains ready to learn. So as we listen to this music, just a disclaimer, I am not an expert of any kind, nor do I claim to be. If anything, I am a master in interpreting scientific data and making it easily accessible to you. And specifically, I like to study genetic data, but really I've been trained in a multitude of ways in order to present information to make it more easily accessible. And obviously am trained in the art of pre-hospital emergency medicine to keep you alive when needed. So those are what I specifically am an expert in. I am not an expert in infectious disease, rather, so do not confuse that. But we will do what we can to divine good information from you from reliable sources, and it will hopefully ease some anxiety, equip you with knowledge, and prepare you for the coming weeks. So, as I said, I love genetics. Genetics is quickly becoming the basis of how we treat patients medically, but also lending to our understanding of how things work. So it's an interesting topic to be able to study and to continue to be part of that research in. What you're looking at here is the coronavirus, and it's interesting how even sometimes the most evil things in the world are beautiful. So this to me is really amazing to look at, but we definitely don't want to get it and look at it within our own bodies. So you'll see that this has a bunch of little spikes all over it, and that's how it infects our cells. So it attaches to one of our cells in our body and makes our cell work against itself. It produces more of the virus within it, and then eventually it allows the virus to get out. This red envelope here, we know that if you use soap appropriately, soap can break this down and actually kill the virus. So that's reassuring in some ways. This in and of itself is something similar to what we've seen before, known as the SARS virus. The technical term for the virus is SARS-CoV-2. So it's a variation of a virus that we have seen before. We do know that there are some mutations with this, specifically within this area where you see the RNA and protein. We know there's a lot of amino acid substitutions there. So amino acids are the building blocks of life. You have amino acids within your body, as does every living organism for the most part. So we know that there's a lot of similarity and dissimilarity between creatures. That's why we're all different. And within this particular virus, we do see this difference right here. And scientists aren't really sure why we're seeing that. It could lend to higher virulence, and maybe it lends to leading a longer life in the summer months. We just don't know yet. It's a little too early to be able to quantify what these substitutions do, but we know that it's there. So these little spikies get into your cell and they infect your cell, and then they begin to produce more of this until they proliferate through. We know that they enter very easily into the respiratory tract. So you breathe it in, it attaches to an attachment called ACE2, which we won't get into. It's a little beyond what the scope of this presentation is, but it attaches there and then begins to make more of it, and then it just spreads through your body. So a virus, specifically this virus, is composed of RNA, and And RNA you have in your body too. So RNA is responsible for protein control. It tells the proteins what to do. But primarily your genetic code is contained within DNA. However, this virus doesn't have DNA, it has solely RNA. So it is going to attach to those places in your body, and then it's going to be able to replicate itself. And because it's an RNA, it's going to begin to mutate proteins. And what we know now, studying on a molecular level, is proteins are really responsible for a lot of the disease processes in our bodies. So this is mutating the proteins at a molecular level, and then that's what's making you sick. Basic difference here is that there's a genetic alphabet that you study. You see the rotation of CGAT, and then in this case you have the RNA, which is replaced, the thymine is replaced with uracil. And then if you want to go deeper, you can study how all these are made up and begin to draw pictures of it in a fascinating study known as biochemistry and organic chemistry. However, if you come away from nothing else, know that we have this genetic alphabet here, and that's what makes up us on a very, very, very basic level. And then these begin to combine together to make up amino acids and so forth. Eventually you get proteins, and then your body begins to build from there. Now this adenine here is important to remember, and we'll go back to it as we talk about some medication options for the COVID disease. Like I said, my job as a bioinformaticist is to look at data and to make comparisons of it and to interpret it for other scientists so that they can make sense of what they're researching. So you don't have to look at this picture and fully understand it. If you look at this, this is just a picture of what the RNA looks like within the virus. Down here is where it gets a little more interesting. So they coded the RNA sequence of the virus and compared it to other viruses that we've experienced before. So we know about the MERS crisis and then the SARS crisis we had in 2003, I believe. Here is our 2019 SARS COVID disease, and it's sitting right here. And this is what you can think of as kind of a family tree. So this is how they all diverge from one common ancestor and how far apart they are. And you'll see this is sitting here in this purple area, and it's most closely related to the bat-like viruses. Now, does it mean that we know for sure that this diverged from the bat coronavirus? We don't know positively. We just know that its sequence is very, very similar to the bat coronavirus. So that could be a hypothesis that needs to be proven eventually. So moving on, you can see these are just another family tree, if you will, of similarity between all of the viruses and how they might differ. And we see here with our COVID that a lot of it's pretty similar on how it's made up. Again, the biggest difference that we saw is in that N protein, but most of it is otherwise up to 90% similarity between the viruses. Here's another image of what we might be investigating when we're looking at all the data that's available to us. This one has actually got an interesting story to tell, though. Again, you'll see the family tree over here. And what this is we as scientists upload genetic data that we get from patients. So you come down with the COVID disease, you get swabbed, your blood drawn, and then we run a genetic analysis on that particular viral genome, and then we upload it to what's called a gene bank and make it available to other researchers so that we can study on how this virus might be mutating, so we can compare it, build these family trees, etc. Now the story here is these two clusters, cluster 1 and cluster 2, are representative of 2 patients in Brazil. So the researchers approached this with, okay, so where did this virus exactly come from? Because as we know from the news and a lot of things that are coming on Facebook, people assume that they all are coming from China. And we know that the spreading of a virus or disease is not nearly as simple as that, or as what we hope to be. So these two, they're in Brazil, right? We can see here that they got it from Italy. They did both travel to Italy and then travel to Brazil and begin to spread it. But then as you go back into this family tree, you see that everything's not clear cut. Remember, orange is China here, and then green is elsewhere. So elsewhere meaning you're going to be getting it from anywhere in the world. And we can compare genetic differences to be able to localize geographically where these came from. So here you do see in one of this particular viral's ancestors, you do see a China, but you also see a lot of the green here. So that means that Italy in and of itself was not just infected by Chinese, it was infected by people coming from all sorts of places. So it's not as simple as China to Brazil or China, Italy, Brazil. It's a little more complicated than that. And you can see here on this enlarged graph that some of the everywhere here is Munich, Germany. You'll see you see Finland, Helsinki. So not as clear cut of a picture as what we hope. And cluster 2 also again shows that as well. You have the patient in Brazil. They had traveled from Italy, but then you get a lot of these greens and no oranges really until you get back to the main graph. And here's the Wuhan genome right there. So there is a lot of divergence with that, and this is showing us that a lot of people from a lot of different countries came into Italy and began to proliferate this virus. So not necessarily solely the fault of the Chinese. Here you see Sydney, Australia, and Paris, France, California. So from really all over, this begins to spread and share. It knows no nationality. It doesn't care where you're from. It just cares that you're human. And we humans are great at sharing things. We've been classically great at sharing stories and breaking bread together for a millennia. So it's not surprising that we're also very good at sharing diseases among one another. And finally, we're going to go back to that adenine that we talked about. So there are 2 hopes for treatment, and there are companies that are working desperately and researchers working desperately to create a vaccine for people to be able to take so that we can slow down the progression of this virus. 2 medications that show a lot of promise for treatment of this. One's called remdesivir and the other one's chloroquine. So chloroquine's been around for 70 plus years. It's an anti-malarial drug, pretty easily accessible. So the researchers included that in this particular study, and you can see this is Wang et al., just released in February, studying these particular medications and their effect on the COVID disease. They were based out of China, so they've been dealing with this since December 2019, whereas some of us have not been dealing with it for that long. So you see chloroquine and remdesivir here, and this is a little molecular zoom up of how they interact with the virus. So the green is your viral load, so you can see that it's pretty significantly in there trying to infect the cells. But then as you introduce more of these medications, you see that that begins to deplete and go away. Really, the most effective that we can see here is the remdesivir. You can see with the introduction of the drug that all of a sudden the virus isn't proliferating anymore. It's not making new of itself. Cytotoxicity is where you'll start seeing, cytotoxicity means the death of cells, essentially, or toxic cells. So this really is the one that we are most hopeful with. But chloroquine, being that it's easily accessible, also has a pretty good curve here in terms of shutting that virus off. Interestingly enough, remdesivir was created to combat Ebola, and now with compassionate use releases, that is, when people are sick with very significant diseases for which we have no cure, regulating agencies such as the FDA here in the US allow early release of clinical trials on humans to test its efficacy. So if we know a person's going to die anyway without treatment, we allow these experimental drugs to be used, with their permission of course, to try to give them hope. So compassionate use is what they've been employing with this remdesivir and chloroquine and any of these other drugs to try to cure them before ultimately death happens. So I mentioned adenine. What this does is it introduces a substitute for that adenine, and it confuses. If you can picture a reader that goes down and reads that alphabet that we were talking about, it slips in there and replaces that A that we have in both DNA and RNA, and it confuses that reader. So all of a sudden, if the reader's confused, then it says, you know what, I'm not going to make any more copies of this anymore. And that's how the virus is ultimately killed off, because if it can't replicate itself, if the reader's not going down that alphabet and reproducing more, then it just dies off. So that is the way this medication works. Pretty interesting on a molecular level. And this about sums up any bioinformatic information that I'm going to give you. Sometimes I feel that it's good to be able to have at least more of a baseline understanding of how things work. It kind of reduces our anxiety about things. And it also gives us a glimpse of not only inside this virus, but inside of ourselves, and allows us to sit with a little more reliable information. But if anything else, looking at these medications and knowing that people are working diligently to create vaccines might give you hope, and sometimes hope is hard to find in times like these. So please join me as we continue with our short series. We'll try to keep it short for everyone's attention span, and I'll try to deliver you information that is not Wikipedia based, but reliable in terms of these are agreed research points that other people are replicating as well to begin to study this virus. Thanks for joining me, and we'll see you next time.